Welcome to this technical guide on how to process samples and measure ATP bioluminescence for any HemoGenix assay. This video carries on from where performing the ATP standard curve and controls left off. Hello, my name is Ivan Rich and I'm the CEO of HemoGenix. There are a few requirements for processing samples to measure cell proliferation or cytotoxicity using HemoGenix ATP bioluminescence assays. The cell samples will have been cultured directly in one or more multi-well plates, and these can be 96 or 384 well plates, provided with the assay kit. Or if cells have been passaged and expanded, they will need to be transferred to one of these plates for processing and measurement. Prior to processing the sample plate or plates, perform an ATP standard curve as described in the assay manual, or in our technical guide video entitled Setting Up the ATP Standard Curve and Controls for all HemoGenix ATP Bioluminescence Assays. Remove the sample plate or plates from the primary incubator and either transfer them to a 23 degrees Celsius incubator or equilibrate at room temperature. You'll need a few things to measure ATP of the samples. First and foremost, for all procedures, always wear laboratory gloves. ATP is present on the skin and can influence the results. You'll need pipette tips and an 8-channel calibrated pipette, preferably an electronic pipette. You'll also need a reagent reservoir wide enough for the 8-channel pipette. The ATP enumeration reagent that has been thawed and brought to room temperature, and of course the plate luminometer. And please don't forget to wear protective clothing and use universal precautions. Now, you may not have used all the wells of the plate for your experiment. No problem. You can maintain the sterility of the unused wells, and to do this you're going to be using the sterile adhesive foil that was included with the assay kit. Place the culture plate in the hood and remove the lid under sterile conditions. Remove the foil from the clear plastic container. Holding the foil with the paper side down, peel off the backing paper. Take care not to touch the adhesive side of the foil. After removing the backing paper from the foil, hold the foil at the edges as shown in the picture. Now place the foil over the plate, making sure that it covers all of the wells. Press down the foil over the whole of the plate so that it adheres. Make sure that the foil is flat over the whole of the plate. With a scalpel or sharp knife, cut the foil around the wells that are to be processed. Now peel away the foil that has been cut around those wells. The remaining unused wells will remain sterile for use at a later date. Now let's turn to how the samples are processed for ATP bioluminescence. Here we have a plate in which 3 samples have been plated at 6 replicates, and it's ready for processing. The ATP enumeration reagent is being used from a previous ATP standard curve that was performed prior to measuring these samples. Since only 6 replicates are being processed per sample, an 8-channel pipette is being used, but has been fitted with only 6 pipette tips. Using the same procedure for adding and mixing the ATP enumeration reagent to perform the ATP standard curve, take up 0.1 milliliters of the enumeration reagent and add it to the first set of wells. Hold the pipette at a slant and keep the pipette tips above the surface of the samples. When the ATP enumeration reagent is added, the color of the sample medium will change from red to orange. It's now necessary to mix the contents of the wells. This is an important step because if the cells are not mixed with the reagent properly, the cells will not be lysed, and this can lead to false ATP values. This diagram shows how the wells are mixed. Place the tips of the 8-channel pipette in the center of the wells and withdraw 0.1 milliliters. Keeping the tips under the surface of the liquid and without moving the pipette, dispense the contents of each tip back into the well. Mix 3 times in this first position. Now move the pipette to position 2 and repeat the process. Repeat this process for all 5 tip positions shown in the diagram. Discard the tips once all the mixing has been completed. Now that the ATP enumeration reagent has been added to all wells and the contents mixed, transfer the plate either to a drawer or to a luminometer and incubate in the dark for 10 minutes. After 10 minutes, bioluminescence production can be measured. From setting up the ATP standard curve and controls to measuring the bioluminescence of the samples should take about 30 minutes. Depending on the instrument, measurement of all 96 wells should take less than 4 to 5 minutes. How are the ATP concentrations calculated? Well, the output of any luminometer is in relative luminescent units, or RLU. This is a non-standardized unit of luminescence. To standardize the output, it is necessary to use the ATP standard curve. As described in the assay manual and in the ATP standard curve video, the standard curve needs to conform to specific parameters prior to measuring any samples. These parameters are provided in the assay manual. Here you see a typical ATP standard curve. There are several ways of converting RLU values to standardized ATP concentrations. The easiest, if available, is to use the luminometer software to automatically interpolate the ATP concentrations directly from the standard curve. The raw results can also be exported to a third-party software such as TableCurve, and the results calculated using this software. The results can also be exported to an Excel file and the results calculated using Excel functions. Please refer to the assay manual for detailed information. This concludes this technical guide. If you need any assistance, no matter how small, please contact us. We're here to help you. Thank you.